Hello, everyone, and welcome to today's webinar, CRISPR-based transcriptional activation tools for silent genes in filamentous fungi. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by GenScript. To learn more, visit genscript.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Laszlo Mozsik, postdoctoral researcher, University of Groningen, Leiden University. Laszlo, you may now begin your presentation.
Thank you for the introduction. Hello everyone, and welcome to my presentation about CRISPR-based transcriptional activation tools for silent genes in filamentous fungi. My name is Laszlo Mozsik. I am a postdoctoral researcher at the University of Leiden, and during this presentation I'm going to talk about my work that I did as a PhD student at the University of Groningen in the department of Arnold Driessen. As a table of content through the presentation, I would like to talk about the importance of filamentous fungi, secondary metabolites that are produced by fungi. I would like to introduce our new fungal toolkit for modular cloning, synthetic biology tools for filamentous fungi, and synthetic tools that we use to create with this toolkit for synthetic transcriptional factors, and as well as the title in the presentation, CRISPR-based transcriptional activation tools. And we will close with a summary and answering questions. In the importance of filamentous fungi, if we want to pick industrially relevant members of fungi, we can think about Aspergillus, Trichoderma, Penicillium, and Eustilago species. These species are known about their organic acid production, as well as proteins and enzymes. And next to that, secondary metabolites, which are not part of the primary metabolism and produced to provide other benefits for the fungi, are secondary metabolites, which are natural products which can vary in structure and function and contain toxins, alkaloids, pigments, and even siderophores. During my work, I wanted to investigate this untapped reservoir of secondary metabolites in filamentous fungi. Why do we think that's another reservoir? So far, we have registered 120,000 species of fungi from the predicted size of 1.5 million. And every single species of fungi is predicted to contain around 30 to 70 secondary metabolite producing biosynthetic gene clusters per every single species. Unfortunately, most of the biosynthetic gene clusters are often silent in laboratory conditions, and regulation is also unknown of these clusters. By that, we think that filamentous fungi is an untapped reservoir of potent chemicals in the form of secondary metabolites. So secondary metabolites come in various forms and structures, and their backbones is often produced by non-ribosomal peptide synthetases, so NRPSs, or polyketide synthases, PKSs, or terpene synthases. For example, important members of the NRPSs are the penicillin, the first real antibiotic produced by Penicillium carcinogenum, Penicillium rubens, and cyclosporine, which is used now as an immunosuppressant, as well as the mycotoxins, aflatoxin, and ionotomes, which are polypeptide synthase based, as well as the lovastatin, which is a cholesterol lowering agent. How can we target biosynthetic gene clusters in filamentous fungi responsible for secondary metabolite production? So the backbone of these compounds, it is produced commonly by NRPS or PKS multi-domain mega enzymes. These protein domains are conserved and can be found by using bioinformatic tools. For example, the anti-smash program can do that for you. And it helps you in the identification of biosynthetic gene clusters by comparing your sequences with database repositories, for example, the myriad, where researchers also share their findings and knowledge about biosynthetic gene clusters. Biosynthetic gene clusters often surrounded by other genes, for example, regulators, transporters, or resistant genes and other tailoring enzymes, which further form the created backbone compound. And this can be also used to identify clusters. Unfortunately, the biosynthetic gene clusters are, as we mentioned, often silent in laboratory conditions, and there are many conventional ways how these clusters can be transcriptionally activated, the promoter of the genes in the gene cluster. For example, different growth conditions or co-cultivation can trigger the promoters of these genes, or heterologous expression moving out these gene clusters and moving them into a host that we are familiar in the laboratory, or promoter exchange from the gene or genes of interest by replacing the original native promoter, which is silent, with a new promoter with a strong transcription rate, which would result in activation of these silent genes. There are also global regulators present in the organisms, which are having a global effect on transcriptional regulation. So repression of elimination of a repressor or overexpression and activator could result in a global activation, as well as local transcriptional factors, which are responsible for regulating the cluster. Overexpression, a positive regulator, could result in overexpression of the cluster, or eliminating a repressor could also result in activation of the transcriptionally silent biosynthetic gene cluster for secondary metabolite production. With the advent of CRISPR genome editing, the filamentous fungal fold, it's also revolutionized genome editing techniques. With the aid of CRISPR-based genome editing, we can now precisely and easily perform gene knockouts, marker-free or marker-based gene knockouts, and gene overexpression through promoter replacement, or it's even possible to integrate complete gene clusters and express them heterologously in a new organism. On the figure, which is presented here, we perform the marker-free elimination of condensation domain of a non-ribosomal peptide synthase, and we only use the 106 base per homologous sequence, marker-free eliminating this unwanted condensation domain. With synthetic biology tools, we can also use, with combination of synthetic biology, we can use new tools for transcriptional activation. Since using synthetic biology, new tools can emerge. For example, modular DNA assembly tools can provide rapid assembly of synthetic transcription units or whole synthetic biosynthetic gene clusters. Or logical gates can be also constructed. For example, we can create on and on switches for transcriptional activation. We can perform a domain swapping in multi-domain mega enzymes using very modular systems. Or, as I'm going to talk about in the presentation, we can use these tools for creating synthetic transcription factors for tunable expressions, or CRISPR-A or CRISPR-I for CRISPR-based activation, and CRISPR interference for transcriptional regulation. So first, I would like to talk about the synthetic transcription factors. So transcription factors commonly build up from a DNA binding domain and an activator domain. The DNA binding domain has the capacity of binding in front of the gene of interest through the promoter, and through the activator domain for transcription activation, or repressor domain for transcriptional repression, can modulate the transcription from the promoter of the gene of interest by attracting the RNA polymerase II or keeping it away for repression. And in synthetic transcription factors, we can create a hybrid of them. So we can combine the DNA binding domain of the choice with the modulator domain that we want to use in our whole system. So by using synthetic biology tools, we can repurpose native biological systems through modular assemblies, and we can construct various synthetic transcription units. And through synthetic transcription factors, combining DNA binding domains and regulator domains, we can modulate the expression of the gene of interest. And this could be tuned on gene of interest by expressing different amount of synthetic transcription factors, or we can modulate the number of upstream activating sequences where these synthetic transcription factors can bind in front of the promoter of the gene of interest, or we could modulate the regulator domain. For example, the strength of the activation domain can also have an effect on the transcription of the gene of interest. To create a synthetic transcription factor for Penicillium rubens, we also aimed to use a native fungal system, repurposing it for Penicillium, and we used the QF regulator from the quinic acid metabolization biosynthetic gene cluster. And in the native system, these quinic acid-based upstream activating sequences, QAF, are present in the promoters of the quinic acid biosynthetic gene cluster. And without the QF positive regulator, there's no transcription coming out of these genes, since the transcriptional activator is not binding to the binding sequences. But when the transcription activator is present, it can bind and activate the gene cluster, and the quinic acid metabolization gene can be transcribed. But in the native system, also the repressor is present, and by that quenching the activation, and there's no activation coming out of this biosynthetic gene cluster. So to remodel this, and it was already done through mammalian cell lines and Drosophila, C. elegans, and mosquito and zebrafish, we repurposed it to Penicillium chrysogenome for taking the activator domain from this biosynthetic gene cluster and the binding sequences. We wanted to create various synthetic control devices using different modules combined together. For example, creating a synthetic transcription factor, combining the DNA binding domain of the previously mentioned quinic acid biosynthetic gene cluster, and combine it with various activator domains. We wanted to test different amount of QUAS binding sequences. Since with the decrease or increase the amount of these binding sequences, we should see a scalable expression through the gene of interest. These QUAS sequences were first with core promoters, or also called minimal promoters. These promoters usually contain TATA boxes. These are GC-rich sequences, and they should be responsible for recruiting the RNA polymerase II to the promoter of the gene of interest. But these are not well defined in filamentous fungi. So for Penicillium, we took around 150 to 200 base pair promoter sequences upstream from the ATG of various genes. And we also tested core promoters from Aspergillus as well as Saccharomyces cerevisiae, and we fused them with the previously mentioned QAS sequences. So this is what we are going to call core promoter to the presentation. And of course, we plan to regulate the gene of interest, and this is for validating created synthetic transcription factor regulated readout. We chose the DS-red as a red fluorescent protein for validation of the system. The synthetic transcription factor as well was also fused with a GFP, so we were able to visualize both the expression of the synthetic transcription factor and the GFP. How can we create a modular system where we always aim to change one module? So when we started to work on this project, we started to use modular cloning for modular assembly of various units. Modular cloning was originally established in 2011 for plants, and then many labs started to use modular cloning for modular assembly, which is using a hierarchical assembly of DNA parts, and rapidly the user can create transcription units consisting from promoters and terminators and various open reading frames or protein fusions in between. And based on this start, we started to build the first building blocks of our modular synthetic biology toolkit, which is now got published at ACS Synthetic Biology, where we deposited numerous promoters, terminators, and open reading frames for creating, for example, synthetic transcription factors, as well as CRISPR-based fusions for genome editing or transcriptional regulation. Using modular cloning, we were able to create synthetic fusions. To introduce the hierarchical assembly first, the user has to create DNA parts that will be cloned into the entry vectors of the system, which are usually contain only one part of DNA, and this can be reshuffled in the next step of the hierarchical assembly. These are called level 1 transcription units, and we also provided in this toolkit a version where we can put this on an AMA vector. AMA1 sequences provide autonomous replication in Aspergillus and other filamentous fungi. So the user can already reshuffle these DNA parts into a fungal replicating vector. And in the next step of this hierarchical assembly, called level 2 final multi-gene vectors, numerous of the previously assembled transcription units can be reshuffled into one big vector. And this, we also provided a vector based where you can replicate these multi-gene assemblies in filamentous fungi. So based on this assembly line, we were able to create numerous multi-gene vectors, which containing synthetic transcription factors, which were ready to use for filamentous fungi. A schematic map of the synthetic transcription factors as you see it on the presentation. So we wanted to test numerous promoters to drive the synthetic transcription factor. So we were able to modularly just exchange these elements in the assembly line, as well as with marked with green, you can see we tested 2 different promoters, as well as 2 different activator domains in the synthetic transcription factors. In the red, you can see that we tested numerous binding sequences for this synthetic transcription factor, 0, 1, 5, and 11 binding sequences, as well as we tested numerous core promoters or penicillium in front of the gene of interest for transcriptional regulation of the gene of interest. And using fluorescent reporters, this system was also easy to validate. That is what I talked about. And the created systems were validated using bioactive micro fermentations, and from the fluorescent readout, we were able to evaluate the expression of the gene of interest, and we saw that based on the chosen corp or minimal promoter, we had different readouts. So if we leave out either the corp promoter or the binding sequences from a synthetic control device, then we got a no expression from the gene of interest, which is a DS thread readout. And depending on the chosen core promoter, we have different readout on the gene of interest, as well as we also saw that the number of binding sequences regulate the expression on the gene of interest. We can see on the slide B that 0, 1, 5, or a 5 reverse sequence, or 11 binding sequences create an increased amount of expression on the gene of interest. And on the slide C, we can see a comparison where we compared two native genes from the penicillin production. So driving PCB-AB and PCB-C, the first two genes of the penicillin biosynthetic gene cluster. And we compared the expression of our best working system, and we were able to show that transcription levels can create comparable level to these highly transcribed promoters. And to show the usability of the system, we also integrated the synthetic control devices into the penicillin biosynthetic gene cluster. So as I mentioned, penicillin biosynthetic gene cluster consists of three genes, the PCB-AB, PCB-C, and penDE. And we integrated binding sequences for the synthetic transcription factor in front of the core promoters of PCB-C and PCB-AB, as well as penDE, where we also integrated the synthetic transcription factor cassette itself, and we put this under the regulation of the synthetic transcription factor. And we saw in everyday where we use 5 or 11 binding sequences for deriving the first two genes, we saw a scalable expression from these genes, as well as since this resulted us to realize that the PCB-AB core promoter is a rather weak promoter, we also built a system where we used a full promoter for driving PCB-AB. And it also resulted that every time when you compare 5 and 11 binding sequences, it results in a scaled expression. So as you can see, synthetic transcription factors-based regulation can result in a scaled expression on the gene of interest, but unfortunately, every gene has to be engineered for this purpose. And CRISPR-based transcriptional regulation, on the other hand, could provide transcriptional regulation without the need for genome editing. For example, if we don't have to introduce CRISPR, but in few words, the CRISPR nuclease originally guided with a single guide RNA to the gene of interest, where it can create DNA breakage, which can be repaired by various ways. If we use nuclease dead Cas9 or dead Cas9, this Cas9 protein doesn't have a nuclease activity, and by that, it still can be guided by using a single guide RNA to the locus of interest, but it does not cut the DNA anymore. And if we are talking about, and if we fuse a regulator domain to this guidable protein, we can also create a synthetic transcription factor that we could create a guide to the promoter of the gene of interest, and by deploying a regulator protein to these segments, we could create regulation, transcriptional regulation on demand. And we chose for transcriptional activation the DPR, tripartite activator domain, which shown to be functional in numerous species already. And we established it for penicillium. And In case the gene of interest would be a transcriptional regulator, a positive transcriptional regulator of a biosynthetic gene cluster, we would also expect that activation of the regulator could also waken up a whole biosynthetic gene cluster. So we built a vector using the synthetic modular toolkit that we established. We created a vector where we can, which consists of two different fungal selection markers, as well as it's expressing that Cas9 Viper fusion, and it has a single guide RNA plug and play module where we were able to plug in the single guide RNA, which contains the target sequence that we want to target with this protein, and we were able to rapidly clone and deliver it to penicillium, which was the host for our readout. And as a proof of principle, we chose the previously established core promoter system without any binding sequences, because if, for example, one of our very weak promoter, the promoter penDE core promoter, should be a very weakly activatable promoter, but with still the capability of being as a functional promoter. And without binding sequences for the synthetic transcription factor system, this promoter should not express detectable amount of red fluorescent proteins, and through the activation with CRISPR-based transcriptional regulation, we should be able to activate this promoter, which still has functional element as being a promoter, and we should see a red fluorescent signal activated. So through fluorescent microscopy, we investigated strains which were targeting 6 different landing sites in the promoter of this promoter penDE. It's nice to notice that we are talking about an only 200 base pair long promoter, where 30% of it is the established untranslated region of the RNA, the transcript. And we are targeting in front of this sequence with 6 different targeting sequences for CRISPR-A, and from 6 different targets, in 4 cases we were able to detect fluorescent signal. So compared to the native system where we did not have a single guide RNA targeting the DS thread. So after we established a fluorescent reporter readout and the system shown to be successful to activate a silent promoter, an artificial silent promoter, we took a native transcriptionally silent biosynthetic gene cluster for secondary metabolites in Penicillium rubens, or previously established Penicillium chrysogenome, and we chose the macroporin antimicrobial biosynthetic gene cluster, which homologue was identified in Penicillium terrestre, but this cluster seems to be transcriptionally silent in Penicillium rubens in our cultivation conditions, and we targeted the promoter of the putative transcriptional regulator of this biosynthetic gene cluster. And we also engineered a strain where we performed promoter replacement next to the CRISPR-A plasmid-based transcriptional activation. Here the promoter is, the whole promoter is around 550 base per long promoter, and we planned to, and we targeted around every 50 base pair one single guide RNA landing site for that Cas9 Viper on both strands of the promoter. And after transforming these constructs into filamentous fungi on solid and later on liquid media, we performed readout to identify macroporin-related compounds. And in 2 cases out of the 20 different targets, we were able to identify macroporin-related compounds through LCMS. We also realized a dark pigmentation, both with the promoter engineering and as well as one of the CRISPR-based activator strains. And it is interesting to notice here that from these 20 sequences, as we can see that some of them are very far from the putative transcription start site, 12 out of 20 would fall into the commonly accepted targeting window of good targets for CRISPR activation. And unfortunately, only 2 of these single guide RNA spacers were able to activate through CRISPR activation the gene of interest. And after validating on solid agar by macroporin related compound, we also performed liquid cultures where we performed qPCR on the genes of the gene cluster. We investigated the regulator, the MUC-A, which is the first gene of the biosynthetic gene cluster, as well as MUC-J, which is the last gene of the biosynthetic gene cluster. And we show realized activation through the biosynthetic gene cluster, as well as the targeted regulator, as well as the biosynthetic gene cluster, which is regulated by the regulator. As a summary, I would like to summarize that Filamentous fungi is a rich reservoir in the form of secondary metabolite biosynthetic gene clusters for useful natural products. Unfortunately, most of these biosynthetic gene clusters are transcriptionally silent in laboratory conditions, and new synthetic biology tools are needed and required for rapid and efficient interrogation of these gene clusters. For example, modular cloning toolkits can speed up the steps of synthetic biology for assembling new synthetic transcription units. For example, synthetic transcription factors can be created for activating whole biosynthetic gene clusters. But unfortunately, using such synthetic transcription factors, often genome editing is needed, although this could provide scalable transcription, which can be scaled gene by gene. But on the other hand, using CRISPR-based transcriptional regulators for CRISPRa for activation or CRISPRi for transcriptional reproduction, this could provide a genome-editing-free transcriptional regulating system. But so far, unfortunately, empirical testing of these single guide RNAs is needed, since there's no real trustable prediction how can such activator single guide RNAs be designed. I would like to close my presentation to say thank you to the people who being part of this project. Of course, my PI, Arnold Driessen, and my co-supervisors, Roel Bovenberg and myriad, and as well as people who part of this work with me, Mita, Ricardo, Niels, and Karsten. Thank you for your attention, and I am happy to answer and discuss questions.
Thank you, Laszlo, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, if active sgRNAs vary by the targeted promoter in the CRISPR activation approach, how could we ensure transcriptional activation?
Yes, it's a good question, of course. The informatic tools can help you to predict a good target for CRISPRa binding, and it's a general rule that in front of the transcription stuff, it's a safe spot for binding for transcriptional positive regulators. And if we want to achieve transcriptional repression, going further down in this transcription start site, we can create a repression effect. But next to that, unfortunately, there are no, and of course, taking into account on and off-target effects, but next to that, only empirical testing is possible to ensure that we chose a working single guide RNA. The other option that we could do is multiplexing, so delivering multiple single guide RNAs at the same time on the same vector, for example, and that would increase the chances that our selected single guide RNA is actually working for the selected promoter of the gene of interest.
Great, thank you. Next question. What other transcriptional regulation domain fusions would be interesting to test in CRISPR fusions?
Of course, for mammalian cell lines and other organisms, various domain fusions already been tested. DNA methylation is possible. For interesting would be histone deacetylase and histone acetylation that could create global, for example, acetylated genome that was created locally loosened up chromatin that could also contribute for transcriptional activation locally for a gene or a whole gene cluster of the interest, according to my opinion.
Great, thank you. Next question. Could we use tools for high throughput transcriptional activation of transcriptionally silent biosynthetic gene clusters?
Well, it is possible to bring to high throughput fungal transformations. So using automated machines, you could create a library of various vectors. We are talking about maybe one single guide RNA or even multiple single guide RNAs deployed on one vector and deployed it to the organism. And that could bring us to the level where we just feed DNA with these activators, and then we could interrogate these genes or whole gene clusters in a more rapid manner, combined with CRISPR-A technology. Of course, we're talking about then synthetic transcription factors, where we have to engineer the genome. That can be a more tedious way to do so, and that can be also brought to automated levels. But yeah, the user has to decide on which approach to take.
Thank you. Next question. What does the fluorescent reporters help to do?
So we use fluorescent reporters to validate our systems. And for the synthetic transcription factors, we labeled with GFP the synthetic transcription factors to monitor the presence and expression of the synthetic transcription factors. And we use DS-red, red fluorescent proteins for validating the readout from the gene of interest. So when we evaluated the readout of the system, we were able to detect the produced DS-red, and we were able to normalize with the produced synthetic transcription factor because it was labeled with GFP. So all the data were normalized by the produced transcription factor in the cell next to biomass.
Thank you.
And of course, we use DS-red readout from the same system for the CRISPR-A transcription factor activator. So in that case, since it's the same silent synthetic promoter that should have been silent, without activator and with activation, we managed to see DS-red readout. That's what we use the fluorescent reporters for.
Great, thank you. Next question. Do you think it would be possible to assemble the system in vitro and later deliver it into a broader range of target organisms, for instance, by synthesizing ready-to-use ribonucleoparticles?
I think that's a great idea. And we also, when we were troubleshooting the first steps of the system, we also considered that option. But we actually disregarded that solution since fungal transformation and later cultivation is a rather time-consuming process. So that would mean that your activator protein has to be present at the moment of transformation, and meanwhile you get to the state when you are having your readout of your transcriptional activation. And I'm afraid that RNP method would not be present, or its effect would not be present as long to be able to have a proper readout. Of course, it's possible now that, for example, you transform with RNP your protoplast and you try to have your readout as soon as possible after the moment of transformation. But I think it's providing a less stable delivery of the CRISPR activators.
Thank you. It looks like we have time for one more question. How concerned are you about off-target activation?
Well, we didn't really look into off-target effects. Of course, we selected all our single guide RNA spacers manually curated for avoiding off-target binding. But if that would be the case, for example, we can create off-target activation of lethal genes. In a very unfortunate case, then we would probably not get colonies. And we could also create a repressor if the CRISPR activator binds on places where it should not, and again, in lethal places. We should not get colonies, but we didn't really take it into account undesired off-target bindings with this system. With the efficiency that we saw that we had to get the multiple single guide RNAs to receive activation, I think it's also with an off-target binding where your single guide RNA is not a perfect match, it's also highly unlikely that you would produce off-target activation.
Well, thank you, Laszlo. Do you have any final comments for the audience?
Well, synthetic biology is speeding up molecular biology and genetic engineering, so let's keep making molecular machines.
Great. Well, thank you again, Laszlo, for your time today and your important research. We would also like to thank LabRoots and our sponsor, GenScript, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed. Until next time, goodbye.